Hello students, myself Popere KS, Assistant Professor at Raya Shikshan Sanstha's RBNB College, Srirampur. So in last lecture we have discussed about the basic technique of plant tissue culture, about the different types of callus, that is culture formation, different callus form by callus culture, anther culture, pollen culture. So in this lecture now we are going to continuing with media preparation. So media preparation means preparation of the nutrient media, and why there is need of this media preparation. So the plant which we are going to select for the regeneration, it's to be needed some nutrient medium. So by preparing media, so by preparation of this media, we can keep the explant in this nutrient media so that it will get all the nutrients and all the other requirement which are required for plant to grow. So for a plant to grow, it will be requiring certain amount of nutrients, salts, vitamins, then macronutrient, micronutrients, then other various factors like light, pH, humidity. All these factors are important for a plant to grow. Now we are going to provide this all as plant get all this from the nature, that is from in the natural condition, but we are going to study by how this plant is going to get all the things in vitro, that is in the lab conditions, how the plant will get all the nutrient or the requirements for it growth. So this is media preparation. Now first is how we are going to prepare the media. For media preparation there are various factors which are important. So these are the different chemical or nutrient which will be requiring during the media preparation. So first of all there is preparation of a stock solution. Now preparation of a stock solution means they are very important for further media preparation. So firstly stock is prepared and then the further process will occur. So in that the stock solution of this different substances are prepared, that is the different substances are, that is salt, vitamin, amino acids, then growth regulator, sugar, agar and water. So these are the main component which will be present in the media. Now this MS medium, this is also called as Murashig and smoke medium, and this is a medium which is commonly or generally used for the plant tissue culture techniques, for culturing the cell in the laboratory condition like organogenesis, or for all this MS medium is used. It is Murashig and smoke medium. Now next is the MS nutrient stock. So how we are going to prepare the nutrient, the stock solution of the nutrient. So nutrient and vitamins, they are prepared as a stock solution. So the nutrient and the vitamin, all the different components, they are going to be prepared in a stock solution. The stock will be from 20x to 2000x, that much dilution can be done further. Now and these stock solutions of the various components, they are kept at 4 degrees C in the laboratory condition at 4 degrees C. Now we are going to see the composition of the media and the stock preparation. So there are different things which will be requiring. So first is the major salt. Major salt is, now there are different types of major salt which should be present in the medium, in the nutrient medium for plant to grow. So first of all the component, that is, now this is the component, the various component of a major salt, and this is the concentration mg per liter, how much mg will be present in per liter of the solution, and amount of 500 ml stock, that is 20x. So this stock is of 20x. Now first is NH4NO3, this is the major salt. It will be present 1650 mg per liter of the solution, and this is 60.5 g for the 20x stock. Like this, next is KNO3, this will be present 1900 mg per liter, and the amount 500 ml stock will be 19.0 gram. Likewise CaCl2 is CaCl2 2H2O, this is also major salt, then MgSO4 7H2O, then KH2PO4. Now there are various concentration of this major salt are given per liter, that is this much mg will be present per liter of the solution. Likewise, these are for the amount required for 500 ml of a stock, that is 20x. So this much dilution can be done. So these are the amount, that is 16.5, 19.0. These are different for the different salt. So the different salt, that is a major salt, their concentration will be varying, their concentration per mg per liter, and also for the during the preparation of a stock they are going to be varying. So they are different and they are very important for a plant to grow. Now next is, like major salt, minor salt are also important in the nutrient medium, they should also be present. Now the component in the minors, the component are HgBO3. Now these are the minor salt, HgBO3, MnSO4, .4H2O, then ZnSO4, 4H2O, Ki, Na2MO, O4, 2H2O, COCl2, CuSO4. So these are the different types of the minor salt which will be present in the nutrient medium. Now this is the concentration given mg per liter, and this is the amount of 100 ml stock, that is 200x. So stock solution will be of the 200x. Now here HgBO3, it will be present in 6.2 mg per liter, that is 260 mg in a 100 ml stock. Now for MgSO4, the concentration is 22.3, that is per mg, that is per liter, this much mg. Now this is for the 100 ml stock, that is 2230 mg. Likewise for ZnSO4, it is 8.6, and for stock this is 860 mg. Ki for per liter it is 0.83, and for 100 ml stock solution it is 83 mg. For Na2MO, O4, 2H2O, this is 0.25 per liter and 25 mg per stock solution. Now COCl2, this is 0.025 per liter, this much amount will be in the liter, and 2.5 mg, that is for 100 ml stock solution. Now CuSO4, that is 5H2O, this will be 0.025 mg per liter and 2.5 mg, that is for the 100 ml stock. So these are for the per liter, how much concentration will be there, and when we are preparing the stock, that is suppose for 100 ml stock, this much amount will be required. So this is about the major and the minor salt. Like we are adding the minor and the major, we are preparing it as stock solution, or how per liter how much mg will be required. So likewise there are also different component, different vitamins like plant growth regulators, or there are certain antibiotics. All these are also, they are also prepared like stock or per liter how much amount is required. But now we are just going to see the different, we are just now going to see the different components. Here I had not given the concentration per liter or the stock solution, but this will be like for major and minor stock there are certain amount. Likewise this also for preparing a certain amount of media, there will be the certain amount of this vitamins or this different, that is antibiotic, they will be requiring. Now we are just going to see the names which are present in the nutrient medium. Now first MS vitamins, the vitamins which will be required in the media, or we are going to add in the media will be, that is thiamine, niacin, glycine and pyridoxine. So these four, they are important in a nutrient medium for a plant to grow normally. Now means when we are going to grow in the lab condition, we have to add this in the nutrient, that is thiamine, niacin, glycine and pyridoxine. Now iron and EDTA, these are also important in media. So iron particular amount is added in a particular amount of solution, like this EDTA is also added. So and next is mesoinositol. Now this is also important in the nutrient medium, its composition will be means this is also should be added to the nutrient medium. Now after that, now these are the things, that is minor salt, major salt, then MS vitamins, iron, EDTA, then mesoinositol. Now one other factors which are also important, or other things, that is plant growth regulator. So this plant growth regulator, they are also important for the plant to grow. So we have to add this into the medium. So first is BAP, that is BAP, it is benzyl aminopurine. Next is NAA, that is naphthalene acetic acid. It should also be present in the nutrient medium or added to the nutrient while preparing the media. Now that is GAE, this is gibberellic acid. Now TDZ, that is thiadiazuron. Next is 2IP, that is 2-isopentyl adenine, and one more is kinetin. So these are the different growth regulators. Natural condition, it will be present naturally in the plant for plant growth, but during when we are growing the plant in laboratory, that is in vitro condition, we have to add this plant growth regulator to the nutrient media. Now there are certain antibiotics, this will be added to the media because so it will be saving the plant or the explant from contamination by different agent which will be causing contamination like fungal spore or bacteria. So by adding this antibiotic, they are not going to harm the plant. So that is kanamycin or cefotaxime. This will also be added to the nutrient media. Now these are the different things which are added to the media, that is salts, minor and major salt, vitamins, then plant growth regulators, antibiotic, then iron, EDTA, mesoinositol. Now these are the things that should be present in a particular nutrient media for plant to regenerate. Now we are going to see, suppose we are preparing the 1 liter media. So in that there should be macro salt solution, that is the macro salt solution should be 50 ml, then micro salt solution should be 1 ml, then FeEDTA stock solution, that should be 5 ml, then vitamin stock solution, that should be 1 ml, and mesoinositol, that should be 2 ml. So this much amount of concentration of this particular stock solution should be there when we are preparing 1 liter medium. Now next is we have to add 30 gram per liter sucrose and dissolve it completely and then make the final volume to 1 liter. So after adding all this, we have to add 30 gram of sucrose per liter, and we then finally have to make the the volume to the 1 liter. Then what should be the pH? The pH it should be adjusted to, that is 5.8, then add the 8.0 gram of, that is 8 gram per liter of the agar for gelling the media and then autoclave it. So for 1 liter this much amount is required. 300 gram per liter sucrose will be added, and it is final volume is made 1 liter. We have to adjust the pH, that is 5.8, and agar is added, that is for gelling the medium. Because of the agar, the media is going to settle down, it will be in a gel form. And after that, now the nutrient medium, it is prepared. Suppose we have prepared it in a conical flask, then we have to close it with the cotton plug and then wrap it with aluminium foil and then keep it for the autoclaving. So this autoclaving is important for, it is a sterilization step, so that suppose during the mixing process or during the preparation of the media, any contaminant or any infection, if it caused to the media, that can be sterilized by autoclaving it. So after autoclave, then we can use that media for regeneration of our plant. So this is about the media preparation. So there are different composition of the different, there are different amount that should be added of the different component for preparing a media. So this is all. Thank you.